How's it going, class? So this is going to be the sixth lecture of the exam two period. This is going to be the final lecture before you take the exam. So today we're going to be talking about muscular healing and the muscular immune repair cascade. Essentially, I wanted to talk about this stuff because I feel like in a lot of courses we talk about oxygen delivery, we talk about the muscles growing, we talk about adaptation, but we don't really talk about like what is the adaptation, what's occurring, how are our muscles getting bigger? What is actually occurring at the cellular level that is allowing our muscles to get bigger, that's allowing us to be able to produce more force, that'll make us better at our whatever performance we're trying to improve at. So today I want to go over these things. Don't get too dived in on the specifics of this material. I'm trying to give you just more of an overarching thing. I'm going to talk about a few specifics just so you guys can kind of basically understand the step-by-step process or the dynamic process that's occurring. But I'm not going to be asking you like what time do neutrophils invade the site? What are the proteins on the outside of the cells called? What is interleukin 6 and 4 responsible for? I'm not gonna be getting at you with stuff like that. I just kind of want you to talk or just sit there, listen to what's going on here, and just kind of soak in the overall concepts of what we're talking about here. And so, like I said, I want to talk about this stuff because I think it's really, really cool. Okay, I'll share my screen with you. I think it's really, really cool, as well as this is what a lot of the research that I will be doing here at the UCF will be involving, or what at least is involving. So muscular healing. So today the objectives are to learn some of the cells involved in the immune repair cascade, as well as follow these immune cells through diapedesis, which is a process of recruitment, which is very similar to the infection control. So the process that the muscle goes through and repair is very similar to just infection control. And this is something when people ask me, what are you going to do for research in your career, or what are you doing there at UCF? And so essentially what I'll tell them is that the immune repair cascade that occurs when you, let's say, you cut your skin, right? We all know we get cut our skin, the immune system comes in, it cleans it up, and then it repairs the tissue, and then you go on your way. Essentially, the same thing happens with muscular tissue. When you go through some sort of unaccustomed exercise or heavy exercise, you get muscular tearing. You get the disruption of the Z line. You get myofibrillar tearing. You get disruption of the plasma membrane of the sarcolemma of the cells. And so all of that is damage. And the essentially same thing occurs. The immune system comes in, it cleans that up, opsonizes the tissue, and then it repairs that tissue. And while these immune repair cascades are somewhat different, they still have some of the same fundamental stepping stones that we can follow along, and we can better understand the repair cascade itself. And we're going to understand how the science is evolving. And so much of the reason that this has not been taught in a lot of exercise physiology courses, in a lot of medical courses in general, is because this is a constantly evolving field. Only recently have we gotten the technology to begin to look at this immune repair cascade and begin to understand what's occurring, to look at the cells, to look at how they're differentiating, how they're migrating, how they're maturing. All these different things are very, very complex. And so my goal here at UCF and with my research going forward is essentially it's a long-term goal. It's probably more better suited for the end of my career or beyond the end of my career. But I'm hoping that if we can better understand the mechanisms and the overall complexity and cascade of the immune response involved in repairing muscular tissue, that'll kind of be a stepping stone or kind of lay the fundamental, it'll lay the foundational, hit your metaphors, Trevor, that'll lay the foundational groundwork for better understanding muscular healing and then better understanding healing of all populations, whether that be post-op, ligament, tendon healing, neurodegenerative healing, skin healing, all of those things. If we can better understand this cascade, hopefully that'll provide some sort of framework for us to better understand more clinical populations. And so I'm hoping that this nerdy gym bro research that I'm doing will hopefully leapfrog other individuals into a more clinical scenario where they can help a wider expanse of people through any sort of healing. And so today we're going to specifically talk about muscular healing now. And the first thing we're going to talk about is the damage, delayed onset muscle soreness. So this is the soreness that you feel usually from unaccustomed exercise, usually eccentric exercise. And so if you've ever done a day where you do heavy negatives, whether that's curls or squats, and the next day or 2 days later, you feel really, really sore, that is a manifestation of delayed onset muscle soreness. And what that really is you're getting a lot of disruption of the Z lines, you're getting tearing of the sarcolemma, you're getting tearing of the myofibrils, the actin and the myosin. essentially what that is just you damaging muscular tissue. And so we see on the right here, this histogram is nice and tidy. The histology-gram is nice and tidy and uniform. And if we look on the right, there is massive amounts of disruption here. We see lots and lots of muscle damage. We see lots of disruption of the Z lines. We see lots of tearing of the myofibrils and of the cells themselves. All of this is damage that needs to be cleared and then repaired so that you can go through muscular remodeling. And how you feel this is you feel this in the form of pain, swelling, loss of force production. So one of the ways in the lab that we can measure this actually is if you put someone through, let's say you put them through a muscle damaging stimulus. So before they do that, you have them, let's say you draw blood and then you have them do MVICs, or maximal voluntary isometric contraction. And so you can measure how much force they produce. And then you can have them come back 3 hours, 6 hours, 24 hours, 48 hours, 72 hours later, and you can draw blood and you can have them do to look at systemic markers, as well as you can look at functional outcomes like force production. You can have them do the exact same MVIC once more, do the exact same isometric contraction once more, and you'll see that their force production has likely dropped. And this is due to the disruption of the ultrastructure, disruption of the myofibrils that prevents them from producing force. So obviously, if you're damaging the things that produce force, you're going to produce less force. And so essentially, we can quantify this. We can put them through a muscle damaging stimulus, and then we can look at the repair cascade afterwards, and we can quantify that and we can track that. And so the idea is that if we're doing that, we can not only better understand that, but we can find nutritional interventions. We can find all sorts of different interventions, supplement interventions that can promote or augment that healing process. And so, like I said, 24, 48 hours later, you can do things like measuring blood. And so one of the things that our lab does is we do blood draws at these time points. And then you can look at things like creatine kinase and myoglobin. And if you remember what creatine kinase is in the cell, and it's what rephosphorylates ADP into ATP in the phosphagen system. And as you can imagine, since it resides in the cell, the only way that it's going to get outside of the cell is if you're rupturing the plasma membrane of the myocyte. And so if we measure that in the blood, essentially that's a proxy measure for how much muscular damage we're having. If we have more muscular damage, that means more creatine kinase is able to leak out into the blood, and therefore the higher the amount of creatine kinase, the higher the amount of muscle damage that we think we ensued, essentially. So it's not a direct measure, but it's a proxy measure, as well as myoglobin is the protein that's only within the muscle. If there's lots and lots of that in the blood, we can assume that we likely elicited a high muscle damaging stimulus. Or if there's not much in the blood, we can imagine that there's not much muscle damaging stimulus, or that it's clearing very fast. This is a very, very complex and dynamic process, but essentially these are just different ways that we can quantify and look at exercise-induced muscle damage in order to track that process and then potentially augment that process. And what my future dissertation project will be looking at what's called resveratrol. And resveratrol is said to be an antioxidant or a reactive oxygen species inhibitor. And so as we'll talk about with neutrophils here, reactive oxygen species are essentially what are released that help to kind of clear up the tissue. And what the idea potentially is that this resveratrol, which is what's found in red wine, potentially has some sort of protective mechanism, or potentially has some sort of immune boosting mechanism. But we won't go too much into that yet. So the first thing we're talking about here is neutrophils. So neutrophils, these are the most abundant immune cell, and they reside in the blood and in the tissue. They mostly reside in the blood, but when an infection occurs or when damage occurs, they move across the endothelial lining and move into the tissue so that they can do their jobs. They're the first cell to arrive at damage, and they use this migration usually peaks within 24 hours, and they release reactive oxygen species to clear debris. So if you've ever seen somebody do a trade job, if you've ever seen them like repair concrete or repair a wall or repair really anything that has structure, what you really need to do is you don't just usually fill in the gap. What you usually do is you create kind of a bigger hole around it so that you can more uniformly fill in that gap, or it just makes it easier to repair that and make it look more normal. Essentially, what's kind of occurring here with the neutrophils is the same. They're releasing these reactive oxygen species that clear the debris and allow for the remodeling to take place later. As well as these neutrophils engulf and destroy pathogens and debris, and they clear and opsonize the tissue and repair the tissue for repair. So again, you don't need to know the super specifics of what neutrophils do. This is just kind of an overall understanding so you can get the overall picture of what's occurring here. Next thing we're talking about is monocytes. And so monocytes monitor the total body environment. So essentially monocytes are flowing around the blood until they are needed. And they are the next cell to arrive to the location. There's multiple different forms of monocytes. So previously, and for a long time, we thought there was only one type of monocyte, and that was what was called classical monocytes. And they're called classical monocytes because they were the first ones we discovered. And those are pro-inflammatory, which means that they stimulate inflammation. They cause inflammation to occur. They release signals that cause inflammation to occur or further cause inflammation to occur. And then we have non-classical monocytes, which are anti-inflammatory. They send signals to reduce inflammation, to reduce the inflammatory cascade, and to promote healing and remodeling. And then finally, macrophages. So macrophages are involved in all processes of cell repair. So they phagocytose or engulf cellular debris and the dead neutrophils. So the dead neutrophils have been there. Go back here. The neutrophils arrived first. They cleared the debris. They did a lot of damage, and they've begun to die, as well as some of the debris that they have left behind and some of the damage that they have done is engulfed by the macrophages and removed from that area. They produce cytokines, chemical signals that help orchestrate healing process. We're going to find at certain times they produce different chemical signals that produce different reactions, as well as the resident macrophages, the macrophages that are already in the tissue, are going to release chemical signals when muscle is damaged. So we'll talk about the satellite cells in a second here, but the cells that are in the muscle tissue are what are releasing these chemical signals to recruit the immune repair cascade. And there's two types, again, of macrophages, and they should look familiar. M1 macrophages are inflammatory, and M2 macrophages are anti-inflammatory and remodeling. So originally they are inflammatory, but then they shift to anti-inflammatory and promote muscular remodeling around 78 hours. And that should be familiar. What else occurs around 48 hours? Well, that's when DOMS peaks and begins to diminish. So the same thing is occurring here. And this is what I'm talking about. These are the things that I wanted to go over. So you kind of go, oh, there's that snap. There's that click in your head, be like, oh, this is why I'm feeling sore for this period of time. This is why I feel the performance detriments for this period of time. We'll get to that a little bit more in a second here. And then satellite cells. So satellite cells are just cells that are in the muscle tissue just chilling. They're just sitting there, the quiescence, they're resting, they're not really doing anything. When we have some sort of myotrauma or some sort of damage, what they do is essentially they activate and they proliferate, and then they differentiate. So they move along the chemotax along the myofibril. They literally crawl along this little myofibril here. They find the area of damage. They fuse to the damaged myofiber, and they regenerate the myofiber with new nuclei. So this is part of the reason that myofibers or muscle cells have multiple nuclei, is because the satellite cells are joining in. But essentially what's occurring is they're just finding this little damaged area and they're filling it in, kind of like if you were to putty a wall. These satellite cells are filling in this damaged area, and that's what causes hypertrophy. And so that's how you can have the same amount of myofibrils within the body, or excuse me, the same amount of muscle fibers within the body. If we remember, adults don't really grow new muscle fibers. They just make the current muscle fibers they have bigger. And that's what these satellite cells are doing. So they're responsible for differentiating into new myotubules and repairing the muscle fibers. Quiescent, meaning resting satellite cells, are physically distinct from adult myofibers. So they're just sitting here, they're just resting until they find the damage, until they differentiate into myofibrils. And these just essentially reside between muscle fibers. That's where they usually are sitting until they are needed. And so I talked a little bit about diapedesis, but what we're going to talk about now is specifically transendothelial migration. And so we remember that neutrophils and monocytes are in the blood. So we can imagine that this is a neutrophil or this is a monocyte that's in the blood. And what happens is if we have some sort of damage here, not the site of infection, we'll say the site of damage. We have some sort of damage here. There's signals that are being released out into the blood. And that's what recruits these cells. That's what recruits these neutrophils or these monocytes. And what they really do is they cause certain proteins to become expressed on the cell surface that are called integrins and selectins. And these cells are responsible for kind of hooking on to these proteins in the endothelial lining. So we get this signal release. It tells these cells, hey, put your hooks out. You know, we need some help. And so it begins to adhere, or it begins to attach to the cell wall. And so what it actually does is it connects to these proteins and it begins to slow down and roll along the endothelial lining, right? You can imagine that proteins aren't like actual physically movable things. They're not like your hand that you can just think and close. What we really need to rely on is just a whole set of hooks. And so the hooks begin to hook and slow the immune cell down, and then eventually it goes into a state of tight binding or full adhesion, where it's no longer moving, it's adhered to the cell lining, and then it begins to squeeze through the cell lining through process known as transendothelial migration, or also sometimes called diabetesis. Diabetesis is more of the entire process of what's occurring here, but this transendothelial migration is how these cells get into the tissue. And so if this is a neutrophil, it remains a neutrophil, and then it goes in and it clears up all this gunk, then eventually dies out. If this is a monocyte, the monocyte moves into the tissue and then immediately becomes a macrophage. I think I'll talk about that in a second here, but then it immediately becomes a macrophage. And so monocytes aren't really prevalent in the tissue. And this is also what's evolving in the literature, is that we used to think that monocytes came in here and they remain monocytes. But now we're thinking that they actually don't. They immediately become macrophages. And what macrophage they become depends on the signals present and depends on the conformation of the immune cell itself. And we'll get into that a little bit more in a second. But essentially, the idea here is that this process right here requires a ton of proteins. This process right here requires a ton of protein. And so it's very, very hard to track what's occurring because there's so many different parts, and the movement of these cells is so complex. So we're looking at here is the macrophages, and I don't need you to memorize these chemokines here. You don't need to memorize these signals. You don't need to know what IL-4 is, IL-6 is. You don't need to know CD14, CD16 negative. These all is what this essentially is just to show you is that dependent on the signals, different cells will differentiate into different things. And so we talked about before, you have the pro-inflammatory M1 macrophages that cause inflammation. They help to clear the debris and get rid of the damage in the area. And so when the macrophages or the monocytes move across the cell wall, the cell barrier, or if there's just undifferentiated macrophages in the tissue, if they have certain signals, they will differentiate into pro-inflammatory M1 macrophages, which will elicit or which will release these interleukins and these signals right here, TNF-alpha and all these other interleukins, which will cause more inflammation. But at some point, there begins to become a slow shift. At some point, these signals right here become more prevalent. They start to upregulate, and the macrophages begin to shift into M2 anti-inflammatory macrophages. And these release anti-inflammatory chemokines and cell signals. And this is essentially what's going to allow for the cell remodeling or the cell repair cascade to begin. So we have the anti-inflammatory, or we have the inflammatory cascade initially, and then we get a shift into the anti-inflammatory repair cascade. And so again, you can see here, these cells right here are all our non-classical monocytes, and these cells up here are all our classical monocytes. And we can differentiate these based on their proteins. Essentially, classical monocytes have the CD14 receptor, CD14 positive, and they don't have the CD16 receptor. And then these cells down here all have different conformations of CD14 and CD16. You don't need to know that. You don't need to know what those proteins do, but it's telling you that dependent on what different signals are, depending on their original conformation, or depending on the signals that they are receiving, they will differentiate into different cells. And so here, if they're getting this signal, IL-4, which is anti-inflammatory, they will turn into M2-like macrophages. But if they come in contact with interferon gamma, if they come in contact with a protein saccharide or microbials, they will become M1-like. And so what this is really trying to show you is that this process is very, very complex. In the summary, the monocytes move to the monocytes move to the tissue of damage. Depending on what's occurring in the tissue at that time, and depending on the chemical signals that are being released, they'll either differentiate into pro-inflammatory macrophages, or they'll differentiate into anti-inflammatory macrophages. And this pro-inflammation, this inflammatory cascade, peaks at about 48 hours. And that sounds familiar. Why? That's where your DOMS peaks. The inflammation that you feel is due to the immune system causing more pro-inflammation and causing more inflammation. At some point, you begin to shift towards M2, you begin to shift towards the remodeling process, which is what is going to lead to the functional repair. And what we know is if we have, let's say we have mice and we knock out this response, we knock out these monocytes right here so that they can't become M2 macrophages, you'll get the initial pro-inflammatory response that you would see, but then you won't get the remodeling response, and essentially you'll just have damaged muscles sitting there, and the mice will get weaker, and their muscle tissue will begin to deteriorate. So we know that these two processes work in tandem, and we know that they're both important, and we can look at these processes and say, hey, what's going on? What are some weaknesses? What are some things that we can strengthen or augment in order to change this repair cascade going forward? And so essentially what I wanted to show you with this is that this is a very dynamic process. The satellite cells release signals that activate macrophages and attract neutrophils and monocytes. So we have satellite cells and macrophages that are already in the tissue that sense the damage and release signals that recruit neutrophils and monocytes. Macrophages and neutrophils release signals that cause further proliferation and differentiation of satellite cells. So there's some perpetuation. Once they start to send signals, they begin to send signals back to each other. When they say, okay, oh, you see we're damaged too? Okay, let's tell even more people that were damaged. And they go, oh, you see that we're damaged too? Let's tell even more people that were damaged. It's the same exact thing. It's the same idea that's occurring there. And what really occurs is, I didn't talk about it back here, but you have your resident macrophages. You already have some of these macrophages in the tissue. So these macrophages are in the tissue, they're doing their jobs. What the monocytes are responsible for is just replenishing these stores. So I said that the macrophages are in the tissue, the monocytes are in the blood. The monocytes are in the blood until the tissue sends signals that the monocytes need to move in and differentiate into these macrophages. I apologize to jump back there, but I feel like that was important. And then the process begins as inflammatory, but then shifts to anti-inflammatory. Classical monocytes turn into non-classical monocytes, and M1 macrophages turn into M2 macrophages. So this entire process is what leads to muscular adaptation and muscular growth. And so I know that was kind of a lot thrown at you, but what I want you to see here is that there's a lot of signals. And this isn't even close. This is 1/100th of all the signals, of all the receptors that are involved in this repair cascade. But what you see here is that there's feedback loops. There's a lot of different other factors that are playing in here. Your vasculature is a major portion of it. Your neurotransmitters are a major portion of it. The autocrine factors, how the satellite cell interacts with itself, how it paracrine, how it interacts with other immune cells, your testosterone, nitrogen oxide in the area. All these things are very, very complex, and they all lead to differing responses. And so this is going to take, what I'm essentially saying is this is going to take a very, very long time to understand. And so understanding inflammation in general is kind of where we've begun. And if you you felt inflammation before in your life, and this is something that I will tell people, I think it's kind of funny, is what people don't understand is the manifestation of all of your symptoms of diseases that you usually get is your immune system doing it to yourself. Like we said here, DOMS, the peak feeling of your soreness is at about 48 hours. That's also the peak feeling of your immune pro-inflammatory response. The muscular damage that's occurring, the muscular damage that you elicited from your muscle damaging stimulus or from your exercise isn't just getting worse over time. The immune system is actually coming in and kind of making it feel worse, and it might be even damaging a little bit more tissue so that I can clear it and get it ready for the remodeling process. But in this way, your soreness that you are feeling is because of the immune response, just like if you got a cold. When you get an upper respiratory infection and you get a stuffy nose, a runny nose, and you get a cough, none of that is what the virus or the bacteria is doing to you. What's happening is, let's say you get a sinus infection. You get bacteria in your sinus that your body does not want. So what it does is it sends a lot of cells there and it begins to make mucus so that you can basically slime, or you can push those bacteria or that virus out. Well, that slime and that mucus causes inflammation of your upper respiratory tissue. And so that's what causes a stuffy nose, as well as that mucus begins to drip down your throat and move into your lungs and cause you to cough. And that mucus moving into your lungs also causes inflammation there. And that inflammation there is what allows you to stimulate your lungs in order to cough. So all of this is a compensatory reaction by the immune system to try to get rid of that bacteria. The bacteria is not causing a runny nose. It's not causing you to cough. What's actually occurring is the immune system's response to the bacteria is what's causing the runny nose and what's causing the cough. If you think about this evolutionarily, the viruses and the bacteria that cause you to cough and that cause you to sneeze are what survive because those are the ones that you spread the most easily. But in no way is the bacteria or the virus actually causing you to do that. That is your own immune system reacting to the pathogen. In the same case here with muscular damage, the soreness that you feel 48 hours isn't just like it's taking 48 hours for you to feel the muscular damage. You were actually feeling the inflammatory immune repair cascade at its peak inflammation, and that is what's causing you pain. That's what's causing a lot of the symptoms of muscular damage. And so overall, this lecture, I want to talk about this because I think it's really, really cool. I think this is a lot of cool information for just understanding basic muscular healing as well as just understanding general health following COVID. There's a lot of social media influence on true health decisions. A lot of different ideas out there, and again, I just urge you guys to stay critically minded and to stay close to your education. There's a lot of individuals out there that pretend to be doctors, whatever it is, a lot of quick get fit quick paradigms, a lot of boost your immune system paradigms, a lot of different essentially lies out there. And I encourage you guys to be very, very skeptical when you hear things like that and understand that the science is complex. It's not black and white. There isn't one simple thing you can do to boost your immune system or to do any sort of thing. It's a very, very complex interaction between all functions, all endocrine, all autocrine, all immune, all neuromuscular, all functions within the body are very, very complex and interact with each other in a very dynamic way. And so basis of this lecture, as I've probably said a couple times, is just to help you guys understand this overall process. Again, you guys don't need to know the very, very specifics of what's occurring here. You don't even know that interleukin 4 is responsible for non-classical monocytes differentiating into M2 macrophages. You don't even know that CD11B is an integrin on the outside of the monocyte that causes strong adherence to the endothelial lining through the ICAM processor. You guys don't need to know that stuff. That's way beyond the scope of this class. Just know the general idea of what's occurring here. Know what the immune cells are primarily responsible for. And when we get to the exam, the quiz is going to ask you a few more specific questions, but when we get to the exam, I'm going to keep it more general because I know that this is out of the scope of a lot of your guys' interests. This is just something that I feel like is missing from a lot of textbooks, and we'll slowly add the textbooks as we get more information and as we get more literature on that. That being said, this will probably be the last lecture that you guys watch before a little review session for exam 2. So I hope you guys really, really enjoyed the course. I was not overly enthused when I found out this course was going to be online. Like I said, I very much enjoy interacting with my students, getting verbal feedback, understanding what's occurring, helping you guys to actually understand the material. And obviously I can't do that as well with an online course. And so this is a bit of a bummer for me to teach. I really, really love the material, and I really, really love the idea that I have a large class and that I might be influencing a lot of you. But the idea that this is online and it's not the best way to learn this material is kind of a bummer for me. And so I really, really hope you guys enjoyed it. I try to make these lectures as lively as possible. I try to do my best to make sure that I'm explaining these things in layman's terms, but also complex enough that you understand them and be able to apply them to your future careers. And with that, I hope you guys actually take the time to do the SBI. So I, as an instructor, I really enjoy teaching. And what I came to UCF for was to do some research, but really to get my degree so that I could go on and I could begin to influence young minds, as cliche as it sounds, but influence young minds so that they can go out and they can elicit real change out in the field, out in the medical field, because I don't get to do that from a teaching standpoint. And so I want to give you guys the basis of knowledge that will allow you to better able spring or leapfrog into your careers and be able to directly affect change on individuals who are going through some sort of recovery or going through disease, and essentially hopefully begin to help there be more positive change in the medical system. So with that, please, please, please fill out those SBIs, the instructor evals. Let me know what I'm doing right. Let me know what I'm doing wrong. I love to hear from my students. If you think I was good, if you think that you like listening to me, please tell me. If you think that I was dumb or I was an asshole or whatever it might be, please also tell me. Please remember that I'm a human being and I have feelings too. So if you really, really hated me, try to be somewhat nice about it. You can tell I suck, but you don't gotta put me in my grave. But just try to be honest. I'm trying to learn and I'm trying to become better for my students so that I can be better in my job and in my career. So I appreciate any constructive criticism you're going to have, or I appreciate any kind words you might have. Those are always really nice to read too. Usually makes my day when I read my instructor evals. So what do you guys have to say? Please say it. With that, this is going to be the last lecture. I'll see you guys in the review session. I hope that you guys enjoyed this course as much as I enjoyed making this course for you. And I really, really hope that you guys learned a lot, and I really, really hope you guys will use this in your future careers, and hope you guys move on to more successful doings. So without that, I'll see you guys in the review session, and you guys have a good day.